My name is Elena Contador. I'm a veterinarian and I take care of fish welfare and health here in our aquabiotech facilities. Just a bit of my background: I graduated in Chile and shortly moved to Canada, where I specialize in fish pathology. I've been around for 16 years working with many species of fish in different countries. And 3 years ago, I moved to Malta to work in Aquabiotech group, gaining much more experience in RAS. Having said that, my presentation will be on fish health management in recirculation aquaculture systems. Before continuing with our presentation, here is an outline of the topics that we will be looking at. First, fish external anatomy, very simple concepts. RAS components, not in detail since other colleagues explain this further. Ammonia and ammonia toxicity and how it affects fish health. Our fourth topic is biofilters. Focus on their function to control ammonia. Then advantages and disadvantages of RAS. And from here ahead, we will be talking specifically about fish health goals, prevention, monitoring and control measures that should be implemented in any RAS facility. Since we will be talking about fish health, I will refer to these concepts quite often, so it's better to clarify them now. Skin, technically called integumentary system, is the tissue that covers the whole fish surface. One of the main characteristics of fish is that their skins form scales, very hard structures that give them extra protection. Gills is the respiratory system. Behind this semicircular structure called opercula, we find the gills. Every time you see fish moving the opercula, means the fish is breathing. The gills then are directly in contact with the water, taking oxygen in and CO2 out. The mouth is the digestive system. So here is where the fish get its food and also drinks water. Freshwater fish drink very little water, while saltwater fish needs to drink constantly to avoid dehydration. Eyes and lateral lines correspond to the nervous systems. Fish get a lot of information through the eyes, but they also count with a system called lateral line, which starts from the opercula to the end of the tail. So if you see these dots all along the fish, those are the lateral line. Here we can see. This system allows the fish to detect water motions and pressures gradients. Have you ever seen fish moving in big groups all at the same time, very well coordinated and not bumping at each other? Well, that behavior is due to the lateral line. Fish will feel other fish through the movements they produce in the water. So now that these concepts are clear, I can introduce you to RAS. As you probably learned in other presentations, RAS stands for Recirculation Aquaculture System. Different components in these systems help to keep an optimal environment for the fish. I won't go further into this since other colleagues approach this topic in a more detailed way. For now, I will tell you that in every RAS there is a culture tank or many culture tanks that held the fish, a drum filter where fish waste or any organic matter coming from the culture tank is trapped, a sump with a pump that collects the water from these two components and push it towards the biofilter. So that's the idea of RAS, recirculate the water over and over again. There are another components as well to help disinfect the water, such as ozone and UV. Oxygen supply is also very, very important. So how a system that recirculates the same water over and over again clean itself. Fish in culture tank are releasing ammonia through the gill, the feces, the urine. So how the system get rid of this ammonia that in high concentrations will become toxic for the fish? In the following slides, I will show you some RAS examples facilities, and also I will explain a little bit of the relationship with biofilter and ammonia cleaning. So here are enlisted different examples of RAS. For intense food production, we have the inland RAS facilities for salmon farms being built, for example, in south of Chile, US, and Norway. Before, this type of facility were only limited to the younger stages of the salmon cycle production. Nowadays is the whole cycle that is being taken inland in order to reduce the impact that the sea cages could have in the ocean. Another examples are public and private aquariums. A lot of them are RAS facilities. Tropical or any pet fish wholesale operation. These are holding facilities that import or export many types of aquarium fish. Finally, research facilities. In here, fish are used to test some feed and vaccines for the aquaculture industry. Or another example is zebrafish studies as human disease models. Before explaining some basic concepts about the biofilter, let's talk about ammonia. Ammonia is naturally found in the environment. It comes from the breakdown of manure, dead plants, and dead animals. It is important to plants, animals, and humans as a source of nitrogen. 80% of the ammonia produced by humans is used as fertilizer. Other applications are to manufacture synthetics, fibers, plastics, explosives, and cleaning products. Ammonia is considered a corrosive substance, and the main toxic effects are usually restricted to the site of direct contact, such as skin, eye, respiratory system, and digestive system. At very high concentrations, it can be fatal. However, many factors can determine whether a person will be harmed or not after ammonia exposition. These factors include the dose, so how much, the duration, how long was the person exposed to the toxic, and how they came into contact with the chemical. Why ammonia is so important in RAS? Fish excrete ammonia through the gills, urine, and feces. As for humans, ammonia can be very toxic to fish. The impact on fish health is also related to the dose, the route, and where the fish is being exposed, meaning what tissue is in contact with the toxic. As you could clearly see in this example, the poor fish, as the ammonia is released from their own body, the ammonia dissolves in the water, meaning that if this ammonia doesn't go away, it will harm the different systems, gills, skin, digestive system, eyes as nervous systems, and let's not forget the important lateral line. Now we come back to the biofilter. A biological filter is a tank that contains filter media made from non-corrosive materials such as plastic, fiberglass, ceramic, clay, or rock. this biomedia has a large surface area where a very special bacteria can colonize. The nitrifying bacteria eat the toxic ammonia, producing nitrite as the final product. Let's explain that better in the next slide. But before, take a look to this surface. This is a biomedia looking under the microscope 5 times, and these slimy things that you see in the surface are bacteria colonies. The nitrogen present in ammonia mostly comes from the protein in fish feed. So the ammonia excretion will increase after feeding. The highly toxic ammonia will be processed by nitrosomonas. This special bacteria lives in the biofilter and convert ammonia to nitrite, which is still toxic for the fish, but not as much as the ammonia. After, there is another type of bacteria, the nitrobacter, also living in the biofilter. This bacteria will process nitrite to nitrate. So nitrate, it's mildly toxic compared to nitrite and ammonia. Nitrosomonas and nitrobacter will be also colonizing other parts of the RAS system, but the biofilter is where they have much more growth surface. Thus, it's important to have an optimal condition in water current and oxygen for these bacteria to do their job in the biofilter. For more details on how to start, maintain, and disinfect a biofilter, see more presentations of our team. So we already know the RAS components and why it's important to keep down the ammonia for the fish health. Now I will show you the advantages and disadvantages of RAS, first starting with the good things. Number 1, a correctly designed and operated RAS requires a minimum daily water intake, enough to clean the filter residues and to replace the water lost by evaporation. Number 2, farmers can control the water temperature and with this the growth rates of the fish stock. Also, RAS farms can produce fish that are not endemic of the area thanks to this advantage. Number 3, the third advantage of RAS system is that they require far less surface area due to the much higher stocking densities. Growth can take place year-round, which maximizes production and allows rapid product turnover. The fourth advantage of RAS related to water is quality control. By maintaining the dissolved oxygen at optimal levels, this technology ensures that fish have better feed conversion and reduced stress, resulting in improved resistance to diseases, less food waste, and higher growth rates. Number 5, fish farming in enclosed facilities guarantees excellent protection against adverse weather conditions and predators. By being able to control the breeding conditions all the time, the fish farmer no longer has to worry about a cold wave killing the larvae or the low oxygen levels due to algal blooms during the summer. Finally, number 6, control discharge of waste. Remember, nearly all the water introduced into the system is reused, means less waste is produced compared to traditional systems. Now we continue with some of the disadvantages. So, as number 1, high energy consumption. The necessity for electricity and oxygen 24/7, especially in high biomass systems. The second disadvantage is increased maintenance. RAS requires more time and care than other systems. Number 3, high upfront investment in materials and infrastructure. 4th, high operating cost, a need for highly trained staff to monitor and operate the system. Following, in case of pathogen infection, the whole system must be emptied and disinfected. A very short response time in case of technical human failures. And finally, as the opposite of many people believes, RAS don't eliminate fish diseases. So after this big introduction talking about RAS, biofilter and ammonia, we're going to start with fish health and what we fish health specialists need to direct our efforts. But before, we have to remember that fish health and welfare is not only a veterinary job, it's a company job, since staff and environment are also involved. In this slide, I'm showing 3 principles of the fish health goals. First, happy fish, meaning we don't want a fish only to survive. We want to give them the best environment they can get. A happy, healthy fish will be less susceptible to get sick, or it will be in a better condition to overcome any disease. Happy staff. People who work with animals need to be comfortable with the workload, schedule, and work environment. It's more likely a happy staff will treat animals better than someone that is unhappy. At the time of planning a business which involves live animals, companies should take this in account. Investment in staff welfare impacts animal welfare in a positive way. Care for the environment. Let's not forget that we live in a gigantic aquarium and our resources are limited. Everything we dump into the environment will come back to us at some point, affecting animal and human welfare. The earth don't have a biofilter. So, constantly updating our protocols, technologies, to find ways to reduce our environment impact is a long-term investment. Now, thinking in what I just said, maybe our biofilter or type of biofilter are the trees, the forest, right? They consume the CO2 that we released and they exchange this to O2, to oxygen. So, yes, we do have a giant biofilter as well. But again, we need to take care of it. What a fish health specialist or a fish health veterinarian should be taking care of. These are the 3 bases of a fish health plan. The most important being prevention and monitoring. We never want to get to the point of controlling a disease. Prevention means planning for long term. Many facilities are designed with very little thought in preventive medicine. The first component of prevention is species biology. When building a RAS, we need to know details about nutrition, water quality, and light requirements of the species we want to hold. This is directly linked to husbandry care, so a lack of knowledge of the species biology could end up in non-infectious diseases. Biosecurity is the second component of preventing diseases. It helps to prevent the introduction of microorganisms that can cause serious diseases in our fish population. These microorganisms are called primary pathogens, and they could be many species of viruses, bacteria, parasites, and fungi. The species biology will tell you what type of water is needed, so it will give the information for finding the most proper site of construction for the RAS. Having said this, we need to decide first what species or species we want to hold in our facilities. Each of them would have different requirements for salinity and water temperature. Are we going to focus on fresh or saltwater species? Cold or warm water species? Do we have easy access to the type of water required for our fish in the chosen site? Believe me, sometimes that is not very well thought before the designing. What about pollutants, such as heavy metals? Is there mining activity or volcanoes close to this area we want to build and take the water from? What is the purpose of our facility? Are we going to produce fish for food or entertainment? What are the life stages we want to hold? Are we going to only hold eggs and juvenile fish? Or maybe we want a growth out facility where stocking densities and feeding intensity is going to be higher than ever in the fish cycle. Are we holding fish able to eat pellets, or we need also live feed such as crustaceans? So why is it important to know the purpose of the system? For each of the different purposes, the fish are in different stage of development. Based on this, we could design the facility especially to fill the physiological requirements of the fish species chosen. A good designing will help to prevent husbandry problems that could end up in non-infectious diseases. In some facilities, the purpose of the system is only one. In other type of facilities, could be multiple. In this slide, I'm showing you the different purposes that I will explain quickly one by one in the next slides. Broodstock system, hatchery, grow-out system, display system, and temporarily holding systems. So we start with broodstock systems. This is basically adult fish for egg production. Broodstock management involves manipulating environmental factors surrounding the broodstock to ensure maximal survival, enhance gonadal development, and increase fecundity. It's also utilized to increase the number and quality of eggs produced and control the timing of maturation and spawning. Farms consider whether they grow their own broodstock or obtain the fish from natural populations. The advantages of growing their own broodstock is that the fish can be selected for traits such as fast growth rates or disease resistance over generations to produce more strong fish. This ability for genetic improvement of stocks is more efficient and produce higher value stock. In this stage, very few diseases would kill an adult strong fish. However, they can be subclinical carriers. Our second purpose is the hatchery. Here we hold eggs that we obtain from broodstock facilities. Important parameters are oxygen and water flow to keep the eggs clean and safe from fungal infections. In the case of salmon, they should be kept in darkness, so the staff needs to have easy access to the trays or the eggs incubator. In freshwater facilities, the main disease concern is a type of fungus called water mold or saprolegnia. The staff should be well trained to detect early saprolegnia infections and be able to pick up the infected eggs so the fungus doesn't spread. Growth out system. As an example, farm salmon culture in freshwater hatcheries under controlled temperature and photoperiod on a well-balanced diet reach the smolt stage between 50 and 80 grams within 1 year. Smolts are transferred to sea cages to reach harvest size about 4 kilograms in 10 to 15 months. In the case of RAS, the smolt is not transported to the sea cages. They are kept in bigger size tanks in salt water. Feeding intensity and fish density will increase as the fish are growing, and this type of facility then needs rigorous management of water quality and nutrition. Temporary holding systems, flexible for any type of fish species, easy to clean and disinfect since sanitation needs to be done every time there is a new fish species in the tank. Example of short-term storage are livestock suppliers, like tropical fish wholesale operations, retail pet stores, and research organizations like Biotech. Display systems. The most common example is the public aquaria or private aquaria. They usually are long-term storage, and depending on the facility, all the system purpose might be needed. So the aquaria could have out of display, for example, hatchery and broodstock. Until now, we have been talking about the first block in a fish health veterinary plan. Being clear about what species and what stages of the life cycle will be held in this facility, meaning the purpose, will influence the facility design. Knowledge about species biology will also give information about the type of water and proper nutrition of our fish. So, a lack of knowledge or failure to reach any of these species biology requirements will have an impact in fish welfare and health. Any disease caused for this lack of knowledge is called husbandry disease. Now we will continue with the second part of prevention in fish medicine, biosecurity. Biosecurity refers to measures aimed at preventing the introduction and spread of harmful microorganisms to animals in order to minimize the risk of transmission of infectious diseases. In general terms, these are the basic biosecurity measures applied in any facility that holds animals, not only fish. Compartmentalization means an animal population contained in one or more establishments separated from other susceptible population by a common biosecurity management system. Microorganisms can survive in people's clothing and equipment and be transmitted from one place to another. Adding restrictions to people, fish, and equipment movements is a vital measure in controlling the spread of diseases. Cleaning removes dirt, preparing the surface of an object to be disinfected. Disinfecting will kill or reduce the number of microorganisms, preventing them from spreading. In this slide, we have an example of compartmentalization and people's movements restrictions in a biotech facility. Inside of each room, there is a RAS system independent from one to another, holding different species of fish with different health status. In the first video, we have a technician with blue clothing. He's showing us the green door sign, meaning he has access to this room. In the second video, the technician with red clothing shows us that he cannot access to the room with the green sign. He's only allowed to enter the room with red sign. As a biosecurity measure, he changed his boots. He disinfects his hands with 70% ethanol. And he wears gloves. As soon as these technicians finish to work in this room, he's gonna take out of the gloves and change his boots to come back to the common hall. In this way, we compartmentalize this room. Cleaning and disinfection are a very important part of biosecurity measures in preventional medicine. They complement compartmentalization and restriction of movements. Cleaning a surface simply removes visible debris, dirt, and dust. In the first video, we see one of the technicians cleaning the floor with water using a power wash. In the second video, we see another technician cleaning the biomedia before being disinfected with bleach. Disinfecting a surface, in this case the biomedia, will kill bacteria and viruses population, which it doesn't happen with the cleaning process. The most common disinfectant we use in our facility is 70% ethanol and bleach. So we finish with the first part of the fish health goals, explaining what prevention measures do we take in our RAS facility. The two main are to think about our fish biology, our fish physiology, in order to prevent problems with husbandry. The second part is biosecurity measures, compartmentalization, restriction of movements of fish, people, and equipment, plus cleaning and disinfection. Now we move to monitoring, the second goal of fish health. An effective disease monitoring system is essential to keep track of our fish welfare. We observe fish daily and record their behavior. Any changes in normal behavior could mean an animal welfare issue. As humans, in fish, high levels of cortisol due to stress are associated with disease susceptibility. So welfare problems detected earlier could prevent serious disease outbreaks due to stress. Monitoring also helps to detect infectious disease outbreaks quickly before they spread and become difficult to control. Monitoring will support then prevention and control fish health goals. In here we have a list of the monitoring that we do in our facility: daily water quality checks, daily facility rounds, so we observe fish behavior daily, 3 times a day, mortality checks, so if in any of the systems there are mortalities, we observe them, we describe them superficially and record. If we don't find any specific cause of mortality checking the fish on the surface, we continue with necropsy and laboratory tests. So for monitoring the water quality, there are specific kits that are commercially available. In this picture, it's mentioned ammonia, nitrate, and pH. You can see them there in the picture. Parameters such as temperature and pH are measured with a probe that sends information to a computer system, so this can be monitored every 20 minutes. Monitoring behavior. Aquaculture technicians feed the fish 3 times a day, so they are the most important people observing fish behavior. In this video, it's not feeding time, but Pietro wants to observe the fish reactions towards feeding. We call this feeding behavior. The normality is the whole fish population in the tank goes to the surface close to Pietro's hand, where the food is coming from. A population of fish that does not get excited about feeding time could be a population that is not feeling well. Pietro also observes if there is any dead fish within the tank or if there is any particular fish left behind. If there is one fish wounded, in this case, as we are in a research facility, we need to euthanize this fish with an anesthesia overdose. Why? Because a contaminated wound is a source of bacterial infection and could infect the rest of the population in the tank. Veterinarians should be in close communication with the technicians, always. Monitoring mortalities. PMC means primary mortality classification. It's the action to check the surface and record daily mortalities for every RAS in our facility. The aim of this is to generate data numbers about fin and gill status, cannibalism, superficial lesions related to water quality, and early detection of any abnormal mortality pattern. Monitoring at the lab. Until now, we have been showing you fish behavior monitoring and PMC monitoring, both of which can be performed by any aquaculture technician. For the following monitoring protocols, we need more specific equipment. In the case of fish health, some of this equipment could be taken to the RAS and perform some of the techniques in the same room as the fish. Some others, like PCR, are more complex and need specific laboratory space and higher biosecurity levels. Okay, a necropsy is when a PMC doesn't give us enough information about the cause of mortality or the possible cause of mortality. We need to open the fish. And for this, we divide the fish in sections for recording: head, body, and tail. The first thing to check is the gill. So we open the opercula and we check in both sides what is the gill condition. In here we can find bleedings, congestions. Sometimes you can find parasites as well, but some of them are big enough to see with the naked eye, but some of them we need the microscope. So we proceed to open the fish. We put a paper towel under, so to avoid the fish to slipper too much. And what we are doing here is to create a window for us to be able to observe all the organs inside of the fish. So we can see liver, swim bladder, head kidney. This fish has a lot of visceral fat content. After opening the fish and observe the organs and any changes that could take us to think about any specific disease, we need to take samples from this body. This video I'm showing you the equipment for the samples we want to take. The microscope, so we have slides and cover slides. This will allow us to check the gills under the microscope to look for any parasites or any lesions that is telling us about water quality. For example, we have alcohol to disinfect our tools, media plates for microbiology and bacteriology isolation. We had formalin if we want to take something that's called histology samples, or in the case of human would be biopsy. The last box was samples for PCR. We have our fish, our dissection tools, and we are all set. wet mount sampling. So in here we have the two fish that I showed at the beginning, opening them. What we do, we get a slide and we identify them. We give for any necropsy a case number, which is 22-150 in this case. We put a drop of water from the same RAS system that the fish came from in the slide, and now we proceed to cut a little piece of gill. As I mentioned in the previous slide very quickly, for this test, we can find parasites, we can find gill lesions that are associated with water quality, for example, congestion, hemorrhages. We can find also something that we call hyperplasia, which it's telling us that something chronic, something has been bothering the gills for a very long time. So after we finish, we take the gill, we put the cover slide, and we are all set to look at the microscope. In here, you observe there is the gill tissue. This is called gill arch. So fish has 4 gill arches, and from the gill arches, you have the primary lamella or filamentous coming out of this gill arch, and in a closer view, like in a more powerful view in the microscope, you could see that from the filamentous or primary lamella comes the secondary lamella. So this is a normal gill structure. PCR and histology. So for PCR, we have these little tubes with some buffers and liquids that will help us to preserve the tissues. We have the formalin flask. It's formalin at 4%. In the case of formalin, we need to work in a ventilated room, and as soon as we open the flask, we need to close it. Formalin, it's a volatile toxic, it's inflammable as well, so we need to be careful. What we do first, we took a little piece of head kidney and we put it in the PCR tube. We shake it to make sure that the tissue is in the liquid. Then we're gonna take another piece of tissue. We disinfect first and clean as well any tissue leftovers. And we proceed to take another sample, another piece of head kidney probably. And we put it in formalin. So formalin will fix this tissue, and we will have as a result something very similar to biopsies in humans. The video is showing us a technician working under a biosafety cabinet, a containment device that protects the sample and the technician from any cross-contamination. She holds the fish with the forceps and cut a window as we show it in the necropsy. With a sterile plastic loop, she approached the head kidney of the fish and obtained the sample. Then she strikes the sample in an agar plate. Well done. If the fish is sick for this particular bacteria, we will have growth after 48 hours at 22 degrees Celsius of incubation. After showing you the monitoring system and the lab techniques that support us on this, I would like to explain why we do all this monitoring when fish are behaving and eating normally. So we monitor them because we want to detect early if there is something subclinical. This means that maybe fish are surviving, but they are not happy. So it's affecting fish welfare. For example, we see the entire tank as one animal. If there is one of 5,000 fish dying every day, that means something is not good in terms of welfare. PMCs and population observations are the best to target these problems. Now, when in a 5,000 fish tank we start having a high amount of mortality, maybe more than 1% of the population daily, we apply diagnosis mode. Since this is not a welfare problem anymore, it's a disease problem because we are having high mortalities. First, we need to define if it's a husbandry problem or an infectious problem. Under my RAS experience and the cases I've seen, 90% of RAS diseases are husbandry problems. The biosecurity on RAS is usually very strong. There is good compartmentalization, so it's very hard to have a primary infectious disease. My focus most of the time would be husbandry. Are the fish being well fed? Are the mortalities eaten? Have the feed and oxygen levels been adjusted for the density of the tank? It's something that we need to adjust daily or weekly, depending how much is the growth rate of the species that we are holding. Sometimes that can be forgotten and the fish start eating at each other. What are the water parameters? Ammonia, nitrates, pH, are they okay? So first, I would rule out husbandry problems, and then I will continue with infectious. To understand infectious diseases, first we need to have a clear concept of primary and secondary pathogen infection. So listen well. The primary pathogen infection is caused by a virus, bacteria or parasites that are coming from out of the RAS system. It's an exotic pathogen that causes specific well-known diseases, for example, in salmon, IPN virus, or BKD, bacterial kidney disease. So that's a primary pathogen, a well-known exotic microorganism that comes from outside and infect our fish. It's exotic from our system. A secondary pathogen infection is usually an endemic bacteria that is in the water, pipes, and filters, or even in the normal fish microbiota, which is the bacteria that fish has in their intestine and in the skin. When fish are under stress, for example, due to a husbandry problem, they are more susceptible to this type of infection. On the other hand, a husbandry problem many times give this bacteria an environment to reproduce faster. So when these endemic bacteria increase in number, they could cause secondary bacteria infections. So that is why in our figure we also connect husbandry with secondary infection of fish. Finally, we have a disease event that we could not define the cause. So that's the question mark. This type of events should be recorded in detail for further investigations and consultancy with other veterinary colleagues. After talking about prevention and monitoring, we go to the last of the fish health goals, control of diseases. The monitoring process guides us to define if it's a husbandry or an infectious problem. If it's a husbandry problem, the control should be focused on the system. We need to communicate with maintenance and aquaculture technicians to check parameters, drum filters, pumps, meaning check the well function of all the pieces of the RAS. If we fix the system, the husbandry disease will be solved. In the case that we have an infectious disease, first we need to define if it's a primary or a secondary pathogen. If it's a primary pathogen, we increase first biosecurity measures and we treat it with antibiotic if it's a bacterial disease. We also can euthanize sick fish or eliminate the affected tank if we are thinking in a viral disease. We can treat with some other chemicals different from antibiotic if we diagnose a parasitic infestation. If it is a secondary infection, we go back again to the system. Is the drum filter working well? Is there any excess of organic matter that is allowing this bacteria to overgrowth? Let's check the ozone and the UV. Again, let's talk with maintenance. Are these two systems working? Ozone and UV help to clear and maintain environmental bacteria and organic matter low, so a failure in this equipment could increase bacteria overload in the water. So for all the fish health goals, we register absolutely everything. Water quality parameters observations, PMCs, laboratory results. Why do we need all this data? As veterinarians, we need a database to apply statistical analysis and learn from the results. We could check fish performances by RAS, by families, and we could identify the most common disease problems during the year. Was this husbandry or infectious diseases? With all this data, we can think about what are the risks of the most common diseases and what we can improve in the future. This is called risk analysis and is an important part of fish health management. Every year, my database will get bigger, and this is the chance to improve our fish health management through the years. There is a lot of lack of knowledge of fish disease and welfare. Being able to work with these amazing animals and collect data might one day be useful for research purposes and help other colleagues with questions. Passing fish health and welfare information through a network is very important to help also improving the aquaculture industry in general. And that's it. Thank you very much for listening to my presentation. I hope you like it. If something was not clear, please feel free to email me and we can discuss, no problem. I let you the links about fish welfare, and in this picture I'm in Choclococha Lake in Peru working with rainbow trouts. Bye bye.